Good morning, everybody. It's very nice to meet you here, and it's my pleasure to give this talk on our very popular ACC column, particularly GFC, aqueous ACC column for the separation of the biomolecules. And as you see from the title of the talk, I'll just give you a brief overview of the columns available for the separation of the biomolecules, and I'll touch base upon the common issues which you find all the time during method development and troubleshooting. Next slide, please. Next slide shows the overview how the talk will go. So I'll introduce about the TSKGL different types of columns available. Then what are the factors when we do a chromatographic analysis, and I found this particular one in a LCGC article. I'll just talk about it because it's a very nice guideline for taking care of the column. And slide 7, basically when you start ACC, you expect some features a column should have and how we can maintain those features in the column. That's the particularly for the protein separation and peptide separation. And slide 9 is the overview of the column line, SW column line, which is basically the most popular column line for the separation of the proteins in the application of the monoclonal antibody and so on. And the slides 9 to 14 will cover the column selection guide for the biomolecules in little bit in broader perspective. And then applications, I'll select mostly the applications which is based on the separation of the proteins, monoclonal antibodies, peptides and so on, separation of the monomer from the dimer or aggregates like that. And as you see here, I put the column lifetime in red because that's very important issue. When you spend some $1,700 or so, you need a column which is very stable. So I'll talk about that little more in details. And slide 37 to 41, it's about the secondary interaction, because as you know, the size exclusion chromatography is the only mode of separation where you don't need any interaction. So, but in the ideal world, it is not possible. There are some situations when you will have interaction. So how far we can manage that, and definitely the conclusion. This is just one slide to show that we have a broad range of the particle size available through our inventory, and some of them are in pre-packed column form, and some of them are bulk media. Some of them you can actually get in both bulk media and pre-packed column form. We have from 2 micrometer to 100 micrometer that wide range of particle size. So we have a scope of seamless actually scale up and scale down as you require. This is a puzzle. Anybody, when you are trying to solve the chromatographic conditions best suitable for your separation, you have to solve this puzzle, and that involves ligand, pH, salt type, buffer concentration, temperature, gradient, column dimension, particle. Now it depends on what mode you are using, but even in ACC there are so many factors in this puzzle which you have to solve before you can actually get a clear cut chromatographic separation method. Next slide, please. Now I have listed some of the factors which actually affect the separation for the protein and peptides. Complex mixture is important problem. Low sample concentration is another factor. Sometimes the sample is very costly, you don't want to waste, so you need some column which is very highly sensitive and can separate. And nowadays LC-MS type of application is becoming very important, so we should have some column which is suitable for that type of application. Aggregation is an issue. Aggregation, sometimes inherent aggregation, sometimes it's not inherent. So these are all maybe because of the secondary interaction also. And when we develop the methods, particularly the chromatographic conditions, we have to take care of all these things: types of salt, what is the salt concentration, because we have a very nice database in our inventory where we have developed a lot of applications where we have shown that the salt concentration plus pH, all these actually affect the overall chromatographic separation. Surfactants are many times is needed for the protein separation at some point, and even surfactant is needed sometimes to increase in the separation and better separation for the molecules also. Other derivatives, additives, denaturant, and heat. Everything. Heat is not that much important in SEC, but other things are very important when you are solving that puzzle for the best mode of chromatographic separation. Next slide, please. Now, I got this particular one from LCGC. You know, John Dolan, he is very famous. He writes so many different articles, and one was 10 commandments for the column lifetime. And definitely all of these are very important, and we try to take care of all these as much as possible, and that helps in increasing the column lifetime. So dedicate the column. Particularly dedicate the column is more important when you are using some kind of additives. Using line filters, flush column regularly, guard columns. We always encourage the end users and scientists that please use the guard column, because the guard column actually helps in trapping all those dirty materials from entering into the column. And whether your column is now under pressure from the dirt materials or not, that you can actually figure it out just by change in the pressure, in the back pressure in the system. Avoid the void, control the temperature, retire early. I mean, he is saying that sometimes we think that column should be used forever, but at some point you have to think that no, it's time to retire. Ensure the supply, minimum garbage, and purity is good. So we have actually, if you go through the blog we have in our website tosotalk.com, there is a very nice blog how long the column lasts, and I would request you whenever you have time, just go through the blog. It's a very nice blog, detailed discussion about the pros and cons of the column lifetime is discussed in that particular blog. Next slide, please. Now, when you buy a column, what you expect from that column? There are certain features like rigid solid support, rigid silica support, which we have. We have tested the mechanical strength of our silica beads, high resolution and sharp peak, fast separation, reproducibility, column stability, analytical and preparative size column, which we have, high recovery, and I will show you some slides where we have shown that we have a very high recovery of all the SEC columns or GFC columns we have for the separation of the biomolecules. And the basic idea of using SEC, particularly GFC, is to maintain the biological activity, and so these are the main features we expect from the SEC column, GFC column for the separation of the protein. Now, in the field of protein and peptide analysis, size exclusion column is basically industry's workhorse. Nowadays, particularly in the monoclonal antibody separation field, 99.9% purity is sometimes desired, and we cannot get just by one chromatographic mode, but at the last phase we use the size exclusion chromatography for the separation and quality control of the protein we separate, monoclonal antibody we separate. So another factor, just like aggregation analysis, is also very important, and GFC has a very important role in separating the monomer from the dimer, trimer, and so on. This is just a tabulated form of the different types of columns we have from our inventory, and their parameters like particle size, pore size, different types of standards we have used to calibrate that particular column. We have used different standards because all proteins are not globular. So some proteins are not globular. So we have used globular, dextrans, globular protein, dextran, as well as PEG, linear, branched and globular. So depending on the situation, depending on the protein interest of your substance, you have to select which calibration curve you have to choose, and we have the different molecular weight range available for each of these columns, and you can select using this table the best column for your separation. Now couple of things, go back please. Yeah, couple of things. I'll talk about the pore size. For example, as you go down from 2000 to 4000, as you see, the pore size changes from low to high, 125, 250 and 450. Now you have seen here there is a column called bioassist column in this particular one. As you know, the SEC column, when comes in steel housing material, there is a situation where you can get the interaction from the housing material itself, and if you suspect that you are getting that type of interaction from housing material, I would request you to go through the bioassist column. They are basically same column, just like stainless steel column, only in the peak hardware. Now if you look at the SW, SWXL and Super SW column line, you see we have 125, 250, 450, and here also 125, 250 and 450, 3 different pore sizes, and in this case we have 2 different pore sizes for Super. Next slide please. So as I said, that the SEC, basic principle of SEC is that the column will separate just on the basis of hydrodynamic radius of the molecule. Now that is the molecular sieving mechanism, which you have to somehow enhance to get the better separation as a function of size, but that's not the ideal situation. So what happens to prevent any kind of secondary interaction, the column is protected by a diol coating, which is kind of proprietary diol coating to shield the protein surface from coming into contact with the column surface from coming into contact with the protein surface and any interaction. Next please. Now as I have shown you that irrespective of SW, SWXL, we have 3 different pore sizes, and for Super SW only we have 2 different pore sizes. We have found that the G3000 column line is the best column line for the most protein sample separation and the molecular monoclonal antibody separation. Next please. Now what is the difference between the SEC columns we have compared to the SEC columns you get from the other vendors? Our column has the more pore volume per unit column volume, which results into the better selectivity and resolution. And as you go from the SW to Super SW, so we have a column line, SW column line. Within that particular column line we have SW, SWXL and Super SW. The difference is the particle size. Particle size decreases from SW to Super SW. Now as you go down the particle size, you know that pressure will increase very rapidly. Now this Super SW generally is used for LCMS type of application, or if you have a very dilute sample or you have a very costly sample, you don't want to waste at all in those cases, because it has higher sensitivity compared to the other columns. Next please. Now this is just one calibration curve. I'm showing here we have a lots of different calibration curves which you can use. As you see, we have used the linear standard, which is PEG, then branch, dextran and protein, globular, spherical, to draw the calibration curve. So right now we will concentrate on the solid black circle. So that is the calibration curve for the protein for G2000, and next panel is G3000 SW, and next panel is G4000 SW column. Now next please. If you look at the middle panel, which is G3000SW column, and if you concentrate of your focus on this particular dot, which represents 150 kilodalton molecular weight, which is the molecular weight in general for the monoclonal antibody, that is exactly somewhere in the plateau in between the total exclusion and total inclusion. That is why this 3000 SW column line is most popular for the monoclonal antibody and protein separation of that molecular weight range. But if you look at the same molecular weight here, it is almost to the total inclusion, whereas here it is total exclusion. That is why G3000 SW or SWXL is the best column for the separation of the monoclonal antibody or a protein of that particular molecular weight or so. Now again, this is just a shorter version so that you can compare your standards linear, branched and globular as a function of different types of column line I have available for the separation. If you know your molecular weight and if you know your nature of the sample, if you know that it's having a molecular weight between 2000 and 70,000, and if you know that this is a branched molecular structure, then you know which column you should use. Next slide please. Now for example, you have no idea what column I have to use because I have no idea what's the molecular weight of it. So we generally request that use the G3000 SWXL column as a scouting column. We have another column which is 15 centimeter column for this, which is QC pack GFC, that is even better, and you can use that for scouting, and then you can come back to G3000 also. Now depending on depending on the elution time, whether it's coming in the exclusion or it's coming in the total inclusion, you have to select either G4000 or G2000 SWXL column. Next slide please. Now what is the difference between the XL column line and the SW column line and the XL column line? In terms of efficiency, if you, this is the y axis where HETP is plotted as a function of the sample load. This is about the XL and this is about SW. In general, SWXL column line, because of the lower particle size, performs much better compared to SW column line. But in terms of loading capacity, they are more or less same. About 250 micro. We generally say 100 microgram, but strictly speaking, even 250 microgram works pretty well without much loss in efficiency. But after that, it starts dropping very, very quickly. Next, please. Now we have done some recovery study as a function of 3 different column lines of XL series, 2000, 3000 and 4000. As I am not going into fine details, but at a glance you can clearly see that even at 100 microgram of the sample load, the overall recovery is very, very good, irrespective of the different types of pore size. Excuse me. We have used the 3 different types of molecules here. As you see, thyroglobulin in this case, it is 78, which is low compared to the others, and the research shows that mostly it's the protein specific, not really column specific. Otherwise, the recovery is very, very quantitative and very high. Next slide please. So this is just again another slide just to show we have different types of column lines available for different types of applications, and we have a selection criteria depending on what sample, nucleic acid, DNA, RNA, oligonucleotide, proteins, it's a large protein or small protein, what kind of protein. Next slide please. Then it's a virus or it's a peptide. You can use this table to figure it out what is useful for you. For virus and very large protein like IgM or so, generally we recommend 6000 or 4,000 or 6,000, something like that. 4,000 is generally used for IgM, virus is 6,000, 5,000 or so. As you see, there is a difference here, PW. So what is the difference between SW and PW? SW means S stands for silica, W means water loving. So from the nomenclature itself, you can figure it out whether it's a silica based column or polymer based column. Mostly the proteins are separated using silica based column. Next slide please. This is a reference I took from a article recently published in this particular journal. It's a very nice article where they have used TSK gel G3000 SWXL column for the separation of the IgG monoclonal antibody, and they have actually induced the aggregation as a function of light, there are so many ways you can actually induce the aggregation. The bottom line of this particular article is that you can use TSK gel G3000 SWXL column to separate the monomer from dimer and the multimers. And I'm not going into the fine details, but they have shown that this is more efficient if you can use two wavelengths. As you see, they have done double wavelength SEC here. They have used 214 nanometer as well as 280 nanometer for this study. But as you see, depending on the type of aggregation method, they are getting different amounts of aggregates, but could be separated very well using this particular column. Now, this is the study we have done in our lab. We have used monoclonal antibody just to see whether the column has any effect on the amount of the monoclonal antibody I am loading onto the column. So we started the blue line. I'm not sure the color is very much clear, but this is blue line is 40 microgram of the monoclonal antibody, and the red is 400 microgram, which is 10-fold higher amount of the monoclonal antibody. And we have tabulated the retention time, efficiency, and asymmetric factor as a function of the amount. And as you see, the retention time is 8.023, 8.061, 4500, 3508, and asymmetric factor 1.22, 1.35. The bottom line of this is that peak efficiency as well as the shape did not change dramatically, though we have loaded 10-fold amount of the monoclonal antibody here. This is the zoomed in figure where you have seen the separation of the fragments and the monomer from the dimer and aggregates. As you know that in the therapeutic separation, therapeutic IgG, you have to report how much albumin protein is there. And so what we have done here, we have added 20-fold excess of IgG compared to the concentration of the albumin, and we tried to find out whether we can separate that and see that. Actually, as you see, this is the human IgG and can be separated from the 20-fold albumin and 20-fold excess of IgG from this particular chromatography conditions, which is very common chromatography conditions for the separation of the monoclonal antibody. Next slide please. Digestion. Digestion is another important thing. So where we have tracked the course of the change of the enzyme as a function of the time when you digest. So we have digested the IgG by pepsine, and as you see, as a function of time, the monomer peak goes down, the other peaks comes up, and they could be very, very nicely separated to the baseline, and you can actually monitor as a function of time the amount of the digested products and the loss of the monomer peak. Next slide please. Pegylation is another important issue for the there are so many factors, like you need a targeted delivery, you need a toxicity related issue you want to avoid. So anyway, we have used 3 different molecular weights of PEG, and we derivatized the lysozyme with that, and red is the position of the 6 lysine residues where it's getting derivatized. and we tried to separate, next slide please, using the TSKJL SWXL column, 3000 SWXL column, and solid line 5 kd, next is 10 and next is 30 of the PEG, and as you see, irrespective of the PEG molecular weight, we could separate the mono from the other mono, di, and the lysozyme peak very nicely. This is the Pegylation. So that shows that this particular column is very nice for the separation of the different pegylated proteins. Next slide please. This is the separation of the peptides using this column TSKJL G3000SW. In this case it is not XL, it is XW column, and we have used a number of the different types of peptides. We have added 0.2% SDS surfactant in this case, and we could separate all of them very well, almost to the baseline, and this is the list of the different peptides we tried. So this is very nice application to show that we have applications without the SDS also for another set of peptides, and if you are interested anytime we can get that from our database. Next slide please. Now can we separate the two peptides differing just by one amino acid? We have done that here using super SW 2000 column, and as you know, the super means it's a 4 micron particle size, smaller particle size. Now next slide please. As you see, peptide 20 and peptide 22, they are differing in only one amino acid. If you look into the chromatographic conditions now, this is not exactly same as we did in other cases. Here we have added 45% aqueous acetonitrile. So it is acetonitrile, containing acetonitrile, and it has 0.1% TFA also, and under that chromatographic conditions actually you can separate the two peptides just differing by only one amino acid. This is a very nice application to show that type of, it's not hardcore SEC in that sense, it has some other factors, hydrophobic interactions and all these things also, but it's a very nice application to show that you can do that. Next please. Now what is the chaotropic agent's effect on the separation? In this case we have separated the fusion protein and we have added sodium perchlorate in the buffer, and as you see, when we used 0.4 moles per liter sodium perchlorate into the buffer, the tailing was improved. You have to follow the green line, which is the green line is the separation using 0.4 moles per liter sodium perchlorate. It definitely improved the tailing. Not only it improved the tailing, it could see more of your aggregate present in that. So again it is done using super SW 3000 column, which is 4 micron particle size column. Next slide please. Recently you have noticed that in LCGC there is a very nice article about the column lifetime, and if you notice that over so many years, 2007, 2009 and 2011, the overall perception of the scientists, end users, have not changed. They think that column to column reproducibility, column lifetime matter more than the price, and that's understandable. And if you compare the total effect of those two over the price, it is 37% against 12, 39% against 14, 34% against 13%. So it's very important that the end users want a column which is stable and it's a good column. So we did some study to see how much is the column lifetime we have in our case, and we have used very simple standard chromatographic conditions first, because that's the chromatographic conditions we use for our quality control. 100 millimolar phosphate buffer, pH 6.7, containing the neutral salt, sodium sulfate and sodium azide, just to avoid any kind of bacterial growth. And we have used the standard SWXL mixture containing 4 different standard proteins and 1 small molecule PABA, and we have used monoclonal antibody in another experiment also. Next please. Remember, when we talk to our scientists, we always request or we always recommend that please use the guard column, please filter the buffer and the sample through the 0.45 micron unit. All these things we recommend, but to give the column extra pressure, we did not use those two conditions in our study, as well as we did not clean the column in between at all from the injection number 1 to injection number 1000. And these conditions, the separations were done using the standard proteins, and as you see, we have we have plotted 5 consecutive runs on top of each other, and we checked one column first whether it's giving the reproducibility over 5 consecutive runs. It is superimposable and absolutely good reproducibility you are getting from this particular column. Now, if we go to the next slide, okay, before that we tried 9 different lots, and we could get exactly very reproducible results, which shows that under these chromatographic conditions this column has no problem. So ideally, if we can take care of other things which can spoil the column, we should expect that column life should be pretty good. Next slide, please. So this is the overlap of the injection number 1 and injection number 1000 of one column. Tomorrow I have a talk on this column lifetime, and that will be the much more elaborated discussion about the column lifetime. But as you see here, starting from injection number 1 to injection number 1000, percentage RSD we calculated for the whole run, n equal to 100, because we collected each and every single data, but when I calculated the RSD, I used every 10th data. So this is very low, very low. I will give you the results tomorrow in the talk much more in detail. But even after 1000 injections, the theoretical plate of PABA was greater than 33,000. It was very stable, and as you remember, the total protein content of this particular single run is 0.6 milligram. So 0.6 milligram I am loading for more than 1000 injections, and still nothing changed in terms of the efficiency. And as you see, the retention time and other asymmetric factors, they are all remaining perfectly same for 1000 injections. Next slide, please. This is G3000SWXL again, because I have shown the standard. Now it's just a protein, and BSA is a very common protein. I have actually got many questions from different end users. Can you separate the BSA monomer from the dimer and trimer, because they use it for different purpose as a standard to begin with. So what I found, I took another column which is already run for 1255 injections using protein standards and other samples, and then I excuse me, run this particular sample BSA, and as you see, the monomer is very well separated from the dimer and followed by the trimer. Next slide, please. Now I compared that particular column which is already run for 1255 injections with a brand new column from competitor S of exactly the same ID, same length, same pore size, and if you look at the resolution between the monomer peak and the dimer peak, the TSKJL G3000SWXL column is giving much higher resolution compared to the column which is the competitor column. Next slide, please. So another question was how consistent in terms of the matrix we are providing inside the column over the years. So many years back in 2005 we did similar experiments. As you read, there are a lot of black and white here, but as you read, in that particular study also no guard column was used. Buffers and mobile phase was not filtered just to give extra stress. Protein samples also not filtered. The column was not cleaned, and in that particular study mobile phase was recycled to give again some more pressure on the column. And as you see, the column lifetime test was excellent, and retention time, asymmetric factor, and the efficiency over 3,000 injections was very good. After that, it started losing because we make a limit of 5% as the, if we cross 5%, we say, okay, now the column is crossing the limit. So after that, it crossed 5%, and we could run up to 4,800 injections, but definitely at that point, column was very low in terms of the efficiency. But what it shows, that the basic matrix, nothing changed over here, nothing wrong, everything is fine. Only thing, depending on the chromatographic conditions and how much care we take in terms of the factors we have discussed earlier in the slides, we can actually use the column for a long time. There are so many factors. Next, please. Now, this is the profile where we have shown the maximum pressure limit for this column, which is 70 bar. And this is the pressure for the column at the injection number 1. And this is the pressure injection number 1000 in this particular study. So over 1000 injections, the pressure did not change much, though we gave extra stress because we did not filter or anything, we did not clean. Now, we compared this data with the study which was done in 2005, and this is the data, the pressure after 2100 injection. Definitely it is 56 bar, which is more, but again it is no way close to 70 bar. One thing I'll tell you, I have visited many scientists and end users. We always request that many times the system pressure is a factor also. We have to be very careful about the system pressure. We have to optimize the HPLC instrument very well by reducing the dead volume, void volume, and all these factors, so that you can actually increase the efficiency much more, even if we have good column. Yes, so the columns never reached and exceeded the pressure in this lifetime study. It never exceeded the pressure in the lifetime study we did so many years back, 2005. Next, please. Now, this is again about the BSA, but it's a different column, just to show that it is extremely reproducible with G2000SW column, and G2000SW column is also very much useful for the separation of protein, monomer, dimer, and trimer for consecutive runs. And also, no splitting of the monomer peak was noticed. Sometimes, next slide please, and sometimes when you overload the column, next please, overload the column, you can get a split peak. But as you see here, we have started with 102 microgram of BSA. The previous slide was 10.2 microgram, so it is already tenfold. And then I compared with the 1.2 milligram, 1.02 milligram of BSA, and even at that high load, anyway it's an analytical column, we are not going to load that much, but still, as you see, there is no problem in separating the monomer peak with the dimer peak, from the dimer peak. It was giving very consistent result over many repeated injections using tenfold higher load compared to the normal injection load we use in an analytical column. Next please. Now about the secondary interaction, which is very important for SEC. There is an article in Bioprocess International which I always request you to read, because this is the article where TSK gel G3000SW column is very exhaustively used for this type of secondary interaction issue, to study that particular issue. I am not going into the details, but this cartoon shows that what happens. A globular protein which is spherical in nature is coming into the contact with the surface of the silica surface. When it comes into the contact, if the secondary interaction is very, very high, then it undergoes a conformational change. And that conformational change, as you notice, because of the conformational change, the binding strength between the silica surface with this conformationally changed protein is so high that it does not come out and you cannot knock it off by any way. So, what they have done, they have used arginine in the buffer, 100 millimolar arginine in the buffer, and found that arginine actually can prevent binding. Now, if you have already loaded the column with the protein and found that the protein totally getting stuck into the column, and then you think, okay, let me add arginine into the buffer, it's not going to work because arginine cannot supersede that binding capacity, binding strength. So, what you have to do, you have to have a column, equilibrate the column with the arginine in the buffer for 10 column volumes, and then you load the protein of your interest, and then you will not find any secondary interaction. It will come very nicely out of your column. And I would request just go through that particular article. It's a very nice article. They have done using UV as well as I think light scattering also. Next please. So, it is not just arginine, we have done many different studies using different types of additives. As you see here, we have done the separation using pegylated proteins, and as you know, secondary interaction is very common or expected with the PEG related proteins, and we have found over 150 injections the retention time is gradually shifting to the higher retention time if you do not have any additives. But if you have additives like 10%, just 10% ethanol in the mobile phase, again you are running 150 injections without any retention shift. And we have found, next please, we have found not just the ethanol, even just BSA can also be used to block all the secondary pockets, secondary interaction sites, and then you run your sample. Now, which additive is going to work best for you, that's a trial and error question. You have to try which works best for you depending on the interaction type. Next please. As you see here, next, as you see here, in this case effect of the surfactant is also shown from 0.005%, here in this case 0.05%, and we have used the non-ionic surfactant to show that that helps in getting the better peak shape and resolution. Next please. And also the recovery. So, in general there are so many different applications we have on this type of interaction, and this is a general guideline particularly about the mobile phase, how we can enhance the molecular sieving mechanism and reduce the secondary interaction, that is by changing the ionic strength. Low ionic strength promotes ionic interaction, and the high ionic strength promotes secondary interaction, hydrophobic interaction. So, we should take care of that and play with that and figure it out what is the best chromatographic conditions for the separation. Next please. But we have found the type of buffer is also very important. We have a full-fledged separation report applications in our database. If you are interested, just let us know, I will send you. But as you see, this is sodium phosphate and potassium phosphate, and this is tris HCl. In general, we have used these three proteins, and as you see, definitely depending on the buffer you are getting the different recovery in terms of separation. Next please. So, now I have already told it, but just to avoid secondary interaction coming from the housing material, I would request instead of using TSK gel G3000 SWXL, which is in stainless steel, use the bioassist, which is bioassist, means is assisting in the separation of the biomolecule, which is basically in the peak hardware, and you should not expect any problem of that type of interaction. Next please. So, the conclusion is like this: silica based, our column is silica based and it is a diol coated to prevent the interaction, is very robust column. No change in the column matrix so far, absolutely no change since the study we did last time in 2005 until now. Lot to lot consistency is extremely good, reliability and dependability of the column is unquestionable, and Since the column was studied without the guard column, and as I already explained everything in detail, that shows that the column is really robust. So, in one line, if I say what is the conclusion? Conclusion is this particular column line is extremely suitable for the separation of the biomolecules in general. Thank you very much, and then I will take your questions.